How do you like that? I was wondering if I was going to get assigned to chair anything. Okay, it's my great pleasure to announce the first speaker, Mihai Pop, who actually is one of my collaborators, one of my favorite people to ever talk to. He's going to be talking about something you're not expecting, because this was not discussed yesterday, Mission Impossible Metagenomic Assembly.
Thank you, everybody. You can hear me okay, I guess? Yeah. I hear myself echo, so it's good. And my title has genome assembly, so I actually believe even genome assembly is impossible. So we'll see how this goes. I do have to say that talking to computer scientists and mathematicians, perhaps this is not surprising, but talking to biologists, they're all surprised that I say crazy things like things cannot be done, and I had to explain to them that we come from a culture where we have a whole field of our science that's trying to prove that things cannot be done. So we're very pessimistic as a whole. Our training makes us very pessimistic. I do want to start by giving the very brief history of where genome assembly comes from, because I read lots of genome assembly papers right now, and most of the references stop in the year 2001 or maybe 2005 sometimes. And I wanted to point out that essentially the first so-called genome assembler was from Roger Staten in the 80s. The first essentially overlap layout consensus, like bona fide genome assembler that actually uses graphs, comes from Esko Ukonen's group in '84. So you have this, the fragments are represented as nodes and the overlaps as edges. And this is essentially what spurred all the work that came out of Gene Myers' lab later on. In '95, Gene Myers said, well, the only way you can really do assembly the right way is to formulate it as a likelihood problem. Essentially find a reconstruction of the genome that maximizes the likelihood that the fragments have been generating from a real genome. The Bruin graph assembly, if you will, comes from Iduri and Waterman in '95. So essentially, we're looking back in the past millennium in terms of where a lot of this work came from, and it's that's missed by a lot of people nowadays. And then there's of course a lot of very recent work, and a lot of people have really contributed to assembly. But most of the foundations, at least the theoretical foundations, have been pretty much established by '95. So to me, when I'm thinking, I'm telling my students, if you think you have this great idea that's completely changed the world of genome assembly, you have to wonder, could all these people who are all very, very smart have missed simple ideas? And I don't think they have. So essentially we have to work really hard to improve things. Simple solutions, other people have thought about them, and maybe they don't work. Although at the same time, the field is changing, so the technologies are changing both in terms of computers and the data that we have available to us. So there is definitely a lot of room for making genome assembly better. Why I'm saying genome assembly is impossible, and this is you can prove this mathematically. So this is a great thing. Essentially, a lot of people think of genome assembly as an NP-hard problem. So this is a genome assembly graph, and what you're trying to find in here is a circle. So this would be a single circular genome, and this is actually a very simple bacterium called Mycoplasma genitalium. Like other simple, essentially it's at some level of perhaps you can see there's a simple problem, but so is a puzzle that looks like this, where you have a bunch of little logs, and there's not a lot of logs, and a dozen or so of logs in a box. You take them out of the box and then try to put them back in, and 3 hours later you're still trying. Okay, so it's the same kind of problem that you're faced with. Everybody knows bin packing type problems are NP-hard, and that's essentially what you're trying to do here. But one thing that's one thing that's essential for a problem to be NP-hard is that you actually have an exponential number of solutions that you have to search through, and you can't really find the right solution without enumerating all the options. And that's exactly what happens here. If you look at the genome assembly graph here, and this is the Mycoplasma genitalium circle, well, there are an exponential number of Mycoplasma genitalium-like genomes you can make out of this graph, and they're all equally likely as far as the assembly is concerned. They're all essentially they're all consistent with the data that we have. Okay, so figuring out which one is the correct one, we pretty much have to try all of the options. Okay, and enumerating all the options is essentially impossible even for simple genomes. And actually a lot of, essentially there's this huge debate, or there was a huge debate over, well, should we do the Bruin graph assemblies or should we do overlap layout consensus assemblies? And this argument was, one of the arguments made was, well, if you use the Bruin assemblers, the problem is a simple problem. It's an Eulerian path problem, or Chinese Postman, essentially a Chinese Postman type problem. And those are very simple to solve in computational terms, essentially. The very simple traversal finds a cycle in this graph. And what was missing from that is essentially once you start saying, I don't want any cycle in the graph, I want the right cycle, that's why the problem becomes difficult, right? Once you start to add constraints to keep the cycle to something that makes sense biologically, if you can encode biological meaningfulness somehow in this graph, okay? So even though some problems can be easily proven to be NP-hard, like this overlap layout consensus approaches can be immediately seen to be NP-hard, so it is true for the Bruin graph assemblers once you take into account additional constraints. So there really is no difference between the paradigms of doing assembly. And in my opinion, the choice of paradigm is really an engineering choice, not so much a choice that actually helps you do assembly better in some way. One thing that, and this is a very controversial term in the theory of information, so I don't want to claim this is true, but there's this concept of conservation of information, where essentially it's like conservation of I don't know, mass or energy and stuff like that in physics. But the basic idea is that as we do various algorithms or processes of the data, we only lose information. We can never gain information. That's of course not entirely true, because if I can, adding random noise essentially creates information, but it's the wrong kind of information, right? So what is happening for us for genome assembly is that we start with something that has all the information that we want, and that's like a single cell or a mixture of cells and so on. And then every single step of the way we lose information. So if I do a sequencing experiment, I've lost information. When I take the data from the sequencing experiment and I feed it to an assembler, I lose information again. Okay? So at the end, what I reconstruct has less information than what I started with. And I mean, this should be quite obvious, even without knowing any of the details, that the information contained in a living organism is completely different from what you get from its sequence, right? But essentially, to me, the real problem in assembly is not so much the kind of the algorithmic questions about how, we're using the Bruin graphs, are we using overlap graphs, are we have the great engineering and stuff like that, but really it's a problem of the information that comes in and what we can do with that information, okay? And I think that's one of our challenges. And I just want to put this up, well, first of all, because we are in the UK, but for those of you who are not intimately familiar with assembly, essentially the problem we have, this is a great example from two of our former students, Adam Philippi and Mike Schatz. But essentially the problem that we have is that if you take a book like The Tale of Two Cities and you chop it up into small pieces, you can't really reconstruct the original text if you chop it up into small pieces, right? So if I have pieces of exactly 3 words, I get a picture that looks like this, and this is what the beginning of The Tale of Two Cities looks like to a genome assembler, okay? So it's not exactly clear what text you're actually reconstructing, right? There's various options where you could say it was the worst of times, it was the age of foolishness, and that makes sense as far as this graph is concerned, but so do many other permutations of this text. So one type of information that essentially is very important to us is the length of the reads that we're given. And just to kind of show you, it's not the most beautiful graph, but essentially the same genome reconstructed with reads of length 50, reads of length 1,000, and reads of length 5,000. Okay, so this is without doing any kind of fancy computer science to try to sort out the graph and so on. It's simply, let's build a graph that is consistent with the genome and see what comes out. Okay? And at 50, you have quite a mess, right? At 1,000, you have a bit of a mess. Essentially, it's something that if you if you like puzzles, you can probably sort out exactly what the genome is from this, right? At 5,000, there's no ambiguity. You have the genome right there, right? So there's a clear indication. The longer the reads, the easier the problem becomes. And when you're talking about, PacBio, 10,000 base pair reads and so on, essentially you don't need people like me to do genome assembly. You can write your own Perl scripts to do it. Okay, so that's at some level the problem becomes trivial there. And we looked at some point kind of what happens essentially. What are the it's very easy for me to say, well, the longer the reads, the assembly is easier, but you kind of, mathematically you kind of have to prove it. And it turns out that's a very nice way to formalize the relationship between the length of reads and how hard the assembly is, and it all relates to how big the genomic repeats are. Okay, so and we are in really there's this spectrum where you can start with reads that are much longer than repeats, right? So this would be this gigantic 10 kb reads that are longer than most repeats in bacteria. So essentially you get a single circle from the assembly, right? In this case, the problem is easy. That's the kind of stuff that you can write with Perl scripts, and it's easy to solve a genome that looks like this. And that's how the human genome was essentially put together by the kind of public effort. There were a collection of back clones. They're about 100 kb long that had been sequenced and assembled separately, and then someone put them together. So it was Jim Kent that spent nights on his computer gluing together these back clones. But it was actually an easy task. I mean, the engineering was a little hard, but the gluing together chunks of 100 kb of DNA, it's not that hard because they span so many repeats so easily. At the other extreme, we have the situation where reads are much shorter than repeats. So that's the picture I showed you, the Mycoplasma genitalium genome, where in that case the reads are shorter than the majority of repeats, and so you have many possible reconstructions of the genome in that case. So essentially, it's not that the assembly is hard, and we can say it's NP hard. Even in that case, but essentially the sequencing experiment does not have enough information to reconstruct the genome, and that's the problem that we have. Okay, so essentially we have lack of information, and it doesn't really matter how smart the algorithms are, how good the engineering is. We're not going to reconstruct the genome. There's no information at the input to reconstruct the correct genome. And then a lot of these kind of NP hard problems actually come in a very narrow range where repeats and reads are roughly the same length, and that's where they're bona fide NP hard problems. But in practice, you don't really see that much. Essentially, those situations don't occur that often. And all of this you can actually prove quite carefully, and it's. And we have a paper in Journal of Computational Biology describing this work. But essentially, if you think about assembly as really the difficulty of assembly comes from this interplay between re-length and repeats, you have to ask what are the repeats, right? And this is where metagenomic assembly gets really interesting, because if I'm looking at a single isolate genome, these kind of green parts are repeats. There's some section of the genome that's repeated in multiple copies. And if you play with bacteria, right, typical, essentially the most complicated thing you generally find are things like ribosomal operons, about 6,000 base pairs in length. Right? If I have a sequence technology that gives me 10,000 base pair reads, then 6,000 base pair repeats are not a problem for me anymore. Assembly is easy. And you can actually see people working with PacBio data. There are a lot of papers that claim to just take PacBio data on a single genome and reconstruct a circle completely computationally. There's no additional experiment. You just get a PacBio data, you get a circle. Okay. This is great, and technologies that give us 10,000 base pair reads are here today, and they're only going to get better. Okay. When we're looking at the metagenome, we have a different type of problem, right? If I can have 3 different genomes that are very closely related. So now the repeat is most of the genome, right? So if you imagine like a mixture in the gut, that's a mixture of different E. coli's, and they're mostly the same genome, but there are some critical differences, and those are the interesting differences, right? If you have the IPAH gene or the Shiga toxin gene and stuff like that in in your E. coli, then it's pretty bad for you. Whereas if you don't have that, you're okay, right? And just those small differences are what are the important things that we're trying to find in a metagenomic data set, right? But that essentially means that we have repeats that are the size of whole genomes. So if I'm trying to resolve those repeats, essentially if I'm trying to do haplotype phasing, first of all, with an unknown number of haplotypes and so on, what I'm looking for are reads that are about as long as a bacterial genome, or definitely hundreds of kilobase pairs in length and so on. So those we won't have for quite a while, okay? Unless someone of you knows of great technology that will give us 100 kb reads. But I don't have any technology that can give us that today, and I'm not exactly sure that we'll see anything in the next 5-10 years. So essentially, in metagenomics, we still have a really serious problem because we are not yet in the position to have enough information to solve the problem. Okay. The other thing that happens in metagenomics, and this is something that's somewhat confusing to biologists. Which is if you don't have enough information, one of the claims is that, well, you just have gaps in your genome, right? I don't have enough reads, I will just have a reconstruction of the genome that has gaps in it. The problem is actually worse because what the genome assembler is going to do is going to take that graph I showed you from the Tale of Two Cities and it's going to realize, well, I don't have these links that were connecting wisdom to best here. So there's one correct path to this graph that kind of makes a lot of sense, and the assembler will just find it and say, well, there's no ambiguity here. Well, there are perhaps a little bit of ambiguity, but it's pretty clear that what Dickens wanted to say was worst of times it was the age of foolishness, right? And this is not what the book said, but that's essentially what the assembler will reconstruct. And if you think even in terms of English, right? If I reorganize the beginning of the Tale of Two Cities, it will make perfect English sense. It won't be the correct thing, but it will make perfect English sense, right? So just knowing English, as very, very well, won't allow you to tell the difference between a correct reconstruction of the Tale of Two Cities and an incorrect one, right? And that's a problem because if I'm looking at a biological string, I can't say, looking at this biological string, oh look, this DNA string doesn't make any sense. Right? Because we know very little about biology in terms of DNA or structure of DNA strings. We'll know a lot less than we know about English. Okay, so if we can't solve it in English, we can't solve it in biology, if you will. Right? So there's a big problem in metagenomic assembly where the fact that you have many genomes that are at some level at the limit of detection in terms of how many reads you can generate from the reads in the assembly, you will not be able to. It's not just that you will get fragmentary information from those genomes, you'll get incorrect information from those genomes, okay? So I think that's an important problem, and you can actually see this. I mean, this is a metagenomic sample. This was oral sample that we looked at, and essentially this is the estimated depth of coverage for the context that we reconstructed, and this is roughly, if you will, the correctness, but essentially it's how much of the reference genome corresponding to those organisms we can reconstruct. And the lower depth of coverage, the less of the genome we reconstruct. And this drop-off is more dramatic than you would expect simply by coverage statistics. And this is because we make a lot of mistakes here as well. So essentially there are contexts that even though they are from something essentially like strep suis or something like that, they don't actually map to the strep suis genome because they have mistakes in them. So whenever I give a talk about this kind of stuff, people say, but we have mate pairs. So it's, you give me mate pairs, I can solve everything nowadays, right? So we looked at that as well, where essentially we tried to figure out, well, how much information do mate pairs give you? And there's a clear use for mate pairs where you have contigs and you link them together with the mate pair information. So now even though the contigs are completely disconnected, now you connect them and you know that they are next to each other. There might be some missing information in the middle, and that's okay. But this view kind of ignores the fact that a lot of the problems in assembly come not from gaps, but from repeats. Essentially, we have genomic repeats that we have that we can't really reconstruct correctly. So you can actually use mate pairs for that, where you can take, for example, you look at your graph, there's 2 big repeats here, these 2 blue nodes in the graph are connected by a mate pair, And if you know the length of the mate pair, you might be able to say, look, there's only one path in the graph that's consistent with that length. And this is a trick a bunch of assemblers do right now. So essentially, just using this mate pair information, you've resolved your genome. Okay? There is a problem with this approach when the different paths, I mean, essentially you're measuring not the DNA composition of those paths, but the length of the paths in the graph. So how big the nodes are or how much DNA is in each of these nodes, right? If you are looking at these red nodes, they're two different paths because of the repeats. They're the same length. You can't tell them apart. Right? That's another issue you run into. And essentially, these mate pairs are completely useless. So there's a lot of, at some level, a lot of people say, well, the longer the mate pairs, the better it is. This statement is clearly not true. If I'm connecting the telomeres of a human chromosome with awesome mate pairs, I still can't resolve the chromosome, even though I have these great mate pairs that are really, really long, right? So, so essentially, what we find, essentially, the shorter mate pairs are more useful because they span the repeat exactly. So now I can say, well, I have to go from green node to green node, and therefore I can resolve this repeat better than using these red mate pairs that are long. Okay. There are a bunch of other kind of technical issues here. If I'm looking at the graph, I'm saying, well, how far apart are these two nodes in the graph? Or can I find a path in the graph that connects these two nodes within a certain distance? That's an NP-hard problem. So essentially, you pretty much have to enumerate all the different paths. When you have lots of repeats, that can become very complicated, right? So essentially, you can run into a lot of technical difficulties using this kind of algorithms in practice. Nonetheless, there are many practical situations where they work. Okay. And we actually played a little bit with, well, what if we can tune the mate pairs to the repeat size, and you can actually get improvements where this is roughly how much of the genome you can reconstruct with standard mate pairs, and here if you tune the mate pairs to the graph, you get a bit better result. I won't talk much more, but it's kind of about this I can talk to you later, but the point is, again, if I have the right information, I can actually get more out of the assembly if I don't. Okay, so it's still this idea that if the information at the input matters a lot more than the algorithm that I'm using. Okay, so so I'll stop a little bit with pessimism right now, right? And I want to focus on the positive because essentially we can't choose what data we get. Well, I mean we can to some extent in the way we design the sequencing experiments, right? But we can't decide we want 100 kb perfect reads. Right, essentially, that's simply the technology does not exist. So, and in fact, we have been able to do everything that we have done in terms of genomics with data that are definitely not good for doing anything with it. Right, so the human genome should not be assemblable, and we have a reference human genome. Right, so there's clearly a lot that we can do with the bad data that we have. And we looked at metagenomic assembly. This was a very surprising result when we assembled the HMP data sets, and then Folco disagreed with some of the assemblies are probably crap, but that's okay. But one very surprising thing when we tried to figure out, can you really assemble with 2 lanes of Illumina a whole gut microbiome, right? So the gut is very, very diverse. And the surprising result was that you can. Essentially, these are, essentially we made rarefaction curves. What if you had, say, only 60% of the sequencing amount that you actually were given? How would your assembly look like at that point? And it turns out that after about 80% of how much sequencing we did, we kind of plateaued in terms of both total amount of DNA. So essentially, if you add more DNA to your assembly, the total size of the assembly did not increase. Essentially, every read fell into a contig that already existed rather than creating a new contig. And measures like, the largest contig and things like that plateaued after about 40%. Right? So essentially, the top of the community we assembled we assembled well enough, I guess, depending on what analysis you want to do. Okay. And this was very surprising because the gut is considered as one of the most diverse communities, at least on the human body, and the results were surprisingly good for me. So you can actually do get something out of these assemblies. The other thing that a lot of people are trying to focus on, well, let's just assemble the genes, right? And this is where we can disagree on whether MGRAS should be using reads or assemblies. but one very interesting outcome of our analysis of trying to figure out essentially how read length affects your ability to reconstruct the genome, essentially how many reconstructions of a genome you could build from read length of length 25 and stuff like that, was that if you look at the genomes, essentially these are plots, is how many of the genomes that we looked at and how many of the genes I find in essentially repeat-free regions. So essentially these are chunks of the assembly graph that are correctly assembled given our data. Even at, I don't know, let's look, well, even at 100 base pairs, like this X, right? This X here, you see that, 95, for almost all the genomes, 95% of the genes can be assembled correctly. Like the genomes might not be assembled correctly, but the genes don't have complex enough repeats to confuse the assembler. And this makes, at some level, biological sense. The same things that confuse the assembler confuse the biology, right? If you have lots of repeats inside of genes, then you can have recombination around those repeats, and then the genes stop working, right? So nature will try to kind of keep the genes somewhat repeat-free. Of course, I'm talking about bacteria, so in larger organisms, you'll find regions in introns and so on that contain repeats, so those would definitely confuse the assembly still. But for bacteria, if you just want to find the genes, you probably are doing pretty well, even though the context might be misassembled, but at least the genes in those contexts will probably be correct. Okay. The other thing that and this has been really successful even for human genomes, right? You can just map things to a reference genome, and as we have more and more genomes, just that mapping gives you a lot of information. So these are a bunch of genomes we extracted from oral samples. We can't really assemble the reference, the actual organism we're looking at, but just by looking at it, we can find out regions that are present in one strain but missing from the other ones and stuff like that. Okay, so it's so just mappings around against reference genomes give us a lot of information that's very useful biologically. There's also, I just want to point out that there's one exciting thing that's happening in metagenomics is that now you have a different type of information that you can use as you build assemblies or do other things, and this is the fact that you don't have just one sample. So one organism doesn't exist as one sample or one isolate. Now this organism exists as this organism in sample 1, the same organism in sample 2, the same organism in sample 3, and so on, right? So the information that you get about the abundance of the organism in multiple sample can help the assembly process. The information that we didn't have before, it's at some level essentially phenotypic information, if you will, right? So if I have many samples with different environmental parameters associated with them, different organisms respond differently to those parameters, so their abundances would be different. So now if you see two contigs that are always correlated in their abundance across many, many samples, they're probably from the same organism, irrespective of whether you have a physical link between them, okay? So this basic idea allowed, this is work that was done in part of the MetaHit project, where they could take all the genes from the MetaHit project and actually cluster them into kind of organism-sized bins, just based on correlation across multiple samples. Okay, and this is a very powerful idea, and we'll see a lot more on this. And there's more and more work going along these lines. So Eric Alm, for example, has an approach to use this information to decide where to cut off OTUs in terms of 16S, right? Do you want a 97% cutoff or a 99% cutoff? He now chooses that cutoff based on abundance of those OTUs across multiple samples, and that's actually a very powerful thing. And I think that there's a lot of stuff that we are not tapping here, right? So each metagenomic sample is 1,000 or 1,000,000 or 100 million biological experiments happening at the same time, right? So we still haven't figured out how to capture that information and essentially take from a metagenomic sample the information about the experiments that were run there and the results of those experiments without running those experiments. I think there's a lot of really cool stuff that that will come out of these studies in the future. All right, so I'm doing well on time, so hopefully there will be good questions. So anyways, I think just kind of brief stop here. Essentially, I mean, I think we can do some things in metagenomic assembly. Essentially, there's clearly room for optimism, and I think there's lots of interesting new things that we'll be able to do because the metagenomic data give us information we didn't have before. But one of the key problems we're facing, right, essentially, I'm telling you, I can't guarantee you that the data coming out of a metagenomic assembly is correct, okay? So the question is, how do we actually figure out if the assembly is right or wrong? And this is a problem that we're facing with single genomes, and any of you who play with genome assembly, you know there's lots and lots of competitions trying to figure out which assembler is better and stuff like that. So I do want to point out that oftentimes we kind of think about errors as kind of like a conceptual error. The assembler is not able to reconstruct the genome correctly and so on. But there's also things like software bugs, and this is one of the first computer bugs ever, and it's actually a bug. And it's actually stored, and it's a museum. I don't remember which museum it sits at, but essentially this is there are lots of computer bugs, and software engineers will give you some number between 15 and 50 bugs per 1,000 lines of code. And if you look at the size of your typical genome assembler, you realize how many bugs are in this code, right? Well, it's any software that you have is full of bugs. And so we have to think not just that, well, the assembler or our idea on how to do assemblies might make mistakes, but also the fact that as you code up that idea, you're going to make mistakes as well. And you have to deal with them at the same time. So how do you do testing? Okay, so this is, you can actually go back to the software engineering literature, and I actually have, I'm working with a colleague, Atif Memon, who's a software engineer, and he works on model-based testing, for example. The typical approach that people use when they do validation of assemblies, and this is like if you do assemblathon, if you look at the gauge competition and so on, essentially you're saying, I have an oracle, I know what the truth is. Okay, so you take the truth, which is in some known genome, you sequence it, you assemble it, and then you ask, is the assembly the same as the truth that you started with? Right? If it's not, any differences are clearly mistakes somewhere. Right? So that's one way of doing it. Part of the problem is, first of all, you don't really know if this difference is due to the assembler or to the way you actually did the sequencing experiment, right? So the sequencing experiment itself can introduce errors in the process that you can't fix with the assembler. Right? The other problem is that different genomes have different characteristics. So just because your assembler does really well on Streptococcus, it doesn't mean it's going to do really well on a metagenomic sample, or it's going to do really well on Staphylococcus. Okay, so how do we deal with this? The other way to deal with this is what software engineers are doing, which are, and one of the ideas is, well, I can model, so this is my assembler that actually builds an assembly, but I can create a model of what my assembler should do, and then see if the result of the model and the result of the actual assembler match. If they don't, there's a mistake in the assembler. Okay? I mean, this doesn't check the fact that our idea on how to do the assembly is correct. Okay? It's because the same idea will be implemented in both of these, right? But it checks whether the software that generated that idea is correct. There's another big problem with this is that if I'm giving you a piece of code that's like 100,000 lines of code and so on, it's not exactly clear to me how I create a mathematical model of what that piece of code is trying to do, and then be able to run that mathematical model and compare the results. Okay, this is very difficult even for simple problems. So the last the most promising avenue forward is this other idea where essentially we're not trying to model the assembler, but we're trying to model the sequencing process. So essentially, I can take my assembly and say, well, if I had this assembly, given what I know about sequencing and experiment, would these reads have come from that assembly if that genome was the correct genome and it was sequenced? Okay. So then I can essentially, I'm comparing, even though I don't know the genome, right, that I'm assembling, but I can ask, is this assembly consistent with the set of reads that I have? Okay, and this is actually a really powerful idea. I mean, this is not a it's not a new idea. Essentially, that's what Gene Meyer said we should be doing in '95, right? And this is what essentially all the statistical machine learning is essentially trying to do. That's how speech processing algorithms work and so on, right? And you can, well, I need to put a little bit of math here, but essentially what you're trying to estimate is well, what we're really trying to find out is the correctness of the assembly. Essentially, what's the probability of having this assembly given the set of reads? And of course, you can now play with the Bayes rule and come up with a different information which says find the probability of a set of reads given the assembly. And that's something we can estimate. And in a very simple case where we don't have any kind of mistakes in the reads, you can actually do combinatorics and get that number. You can actually take into account a variety of other approaches in terms of looking at sequencing errors and so on, and take all of that into account in the probability score. Again, this is not at all a new idea, right? This is what Gene Myers thought is the right way to do it. This is what a lot of these quasi-species assemblers do, right? That's essentially how you reconstruct the population by coming up with these with this score of how well the reads kind of fit our assembly. And I like, essentially Genova introduced one of these ideas of having a single score of how good the assembly is. And I really like that idea because people would say, well, how does this assembly compare to this other one? And I really want a number. I want to say my assembly is a 10, the other one is a 9, so use mine, right? So this allows us to get a single number. It's a likelihood, right? The likelihood our assembly is correct. There are a couple approaches, including one of ours, that appeared recently. They essentially use the same idea. So there's the AL and Seagull. There's extra software you can use to take an assembly and get the likelihood score for your assembly. Okay. What's really interesting when we try to figure out if these methods actually work, because there's a lot of skepticism. So I talked about this with Steven Salzberger, and he said that's a stupid idea. No one would ever want a single score, and that single score won't really mean anything. Okay. What we did here, we ranked the assemblathon assemblies based on this likelihood score, and then we also looked at how the ranking differs for different other measures of assembly quality. So this is the N50 that we all like to use all the time. Says my assembly has a better N50 than your assembly. And what happens here? These are ranked. The different scores of assembly quality are ranked with how close they are to this measure of likelihoods that we used. Okay, so what you can actually see here is that measures that do not use the reference genome. So the N50 does not use the reference genome. So there's no notion in N50 of correctness, right? Those measures that don't use the reference genome are further away from the likelihood score than the measures that actually use the reference genome. So essentially, without knowing what the reference genome, we compute a score that's now consistent with the scores that you get when you use a reference genome and you compare your assembly with the correct assembly. Okay, which means there is information in this score. I won't go necessarily as far as saying if our score is high, the assembly is correct. But by using a likelihood score, you actually do much better than very simplistic measures of contig size. And you don't need the reference genome. So essentially you can apply this approach to genomes you've never seen and take two assemblers or take two different sets of parameters of assemblers and pick the right one for that specific data set. Okay. One very interesting result with this, essentially computing those likelihood scores is really expensive because you need to know where every read occurs in a genome. If you want to tolerate sequencing errors, essentially you have to do an alignment of every read to all the places in the genome where it could match and compute a likelihood score for that read and all the places where it would match. Right? So essentially you have to do a Smith-Waterman. It's essentially a Needleman-Wunsch algorithm of a read against the whole assembly that you need to compute to get the scores correctly. Okay? So this is a very, very expensive procedure. So we thought, well, can we actually get by using fewer reads? Like, do you need all the reads to figure out which assembly is better? So we did a very simple test where we essentially sampled 100 reads, 1,000 reads, 10,000 reads. This is a human chromosome. And it turns out that in our case, after about 10,000 reads, the different scores of the different assemblies converged to what would be ultimately the final score. Okay, so essentially, this is really surprising because I'm saying I have this gigantic assembly that's a human chromosome, and I'm going to take 10,000 reads out of it at random, compute my likelihood score for those 10,000 reads, and tell you which assembly is better. Okay, this essentially is very counterintuitive, but essentially what this means is that the different assemblies are so different that it's enough for me to find one read that's in the wrong place, and I know which assembly is right and which is wrong. Well, not necessarily which is right, but which is better. And also reassuringly the truth. So this is the reference the actual chromosome, right, the correct chromosome, does better than the other measures, right? If it didn't, then we'd have a problem. And you can come up with examples where actually the truth doesn't score as well. So it's essentially when we don't use the right type of information to compute a likelihood score. Right. And you can use this kind of approaches to tune assembly parameters where you look at k-mer sizes and pick the one that maximizes likelihood. And oftentimes it's not the same thing as the k-mer size that maximizes contiguity. So you might get smaller contigs, but they'll be more correct if you use a different k-mer size. So. Right, so there's one thing that's not just so I talked about these likelihood measures of validating assemblies, but there's a lot of work that has been done in validating assemblies for many years, and before folks at Assemblathon or Gage rediscovered them. Right, where essentially you can look at internal consistency measures. You can look at an assembly and say, well, if a bunch of reads have one base pair and the other reads have a different base pairs, then essentially that assembly, there's a problem in the assembly. This is assuming you have essentially you have a haploid genome like a bacterium, right? You can look at situations where May pairs are in the wrong place and say, well, I think there's a mistake here, and so on, right? So all these measures now are slowly getting rediscovered. There's new software to actually be able to find this kind of things and flag the assembly. So it's not just that you get the single score that you would get in our case from the likelihood score, but you can also say, well, in this place in the assembly something's wrong. That also hopefully corresponds to a region of the assembly where you're actually losing likelihood, if you will. Okay. The other thing I want to point out is that we talk too much about the kind of N50 sizes. We pick one number. We like to pick one number. But the shape of the distribution of contig size actually carries a lot of information. So these are, these should really be done in log space, but essentially you can sort the contigs by size and see how much of the genome is contained in contigs of a certain size. And the N50 is the number that essentially hits essentially 50% of the genome, but the shape of the curve tells you a lot of information. And the more useful stuff is to look at the size of the contigs and also look at the errors at the same time. Right, so you might get large contigs, but if you get lots of errors with the same contigs, then you essentially the assembly is not good. And this is it's represented differently, but it's an idea that they came out of Bob Mishra's lab. They call it the FR curve. Right. Just very briefly, essentially, a lot of these things that in terms of validating assemblies with likelihood, validating assemblies by looking for internal inconsistencies and so on, apply to metagenomic data. But there you have to start playing a little bit with how you define your goals. Okay, so if I have a genome that's polymorphic, right? Is it okay that in the common parts I've mixed the two genomes, or is it not okay? Right? You have to make these decisions as you decide how to score the assembly, right? So you can see this as an incorrect assembly of a genome, because the two haplotypes are mixed together in some parts, or you could see it as a reasonable correct assembly because I've separated the divergent parts enough in my assembly. Okay. So essentially I think a lot of the validation approaches have to be focused to the problems you're trying to solve. The one thing that's critical for all of this, as we go to metagenomic assembly, even as we do genome assembly, are standards. Okay. So right now, if I take 3 different genome assemblers, I will get probably 3 different types of outputs that I can't really compare easily. In the old days, I actually could get where every read went into the assembly. I can't get that anymore. Right? We've given up at some point on this idea, and it's very important for us to decide, well, do I really want that information? Should I be forced to reconstruct that information, and so on? All right. And one thing that I'm finding more and more is that we hate standards as a community. So I'm not sure why. And, at the workshop, I asked, well, why don't we have a standard for representing ACGTs and quality values? And people told me it's impossible to have a standard like that. So, right? So we have FASTA, FASTQ, BAM files, and so on. We can't actually agree on one standard. So, and I'm not sure why not, but that's maybe someone would illuminate me. We have extended, essentially likelihood approaches can be extended with metagenomic data, assuming you get the right information. So if the assembler tells you this contig has this multiplicity, then you can actually use that information to compute a likelihood score. that now fits the metagenomic data. Right, and now I finish. I think just to kind of summarize all of this, I think one of the key lessons I think I'd like to pass on, essentially, I think we know a lot about assembly in theory, right? So essentially, I think a lot of the kind of basic assembly approaches have been sorted out. And there's not a lot of room in terms of saying, I'll have a much better algorithm in general to do assembly in general, okay? Where I think there's a lot of, and you know, a lot of it is also this garbage in, garbage out. If our data sucks, the assembly is going to suck. There's no way around that, okay? But one of the important things that we need to do is figure out essentially what problems we're trying to solve, and then figure out how, essentially, formalizing what we want to get out of the assembly. If we want just the genes, then we should have an assembly that just reconstructs the genes. And we evaluate the assembly based on how well it reconstructs the genes, not based on how big the contigs are, right? If we want to reconstruct, for example, in metagenomic samples, a lot of times I might just want one genome after the sample, right? I don't care if my assembly looks good overall. I just want to know that this pathogenic strain that happens to be my sample and I can't culture it, that strain is assembled correctly. I don't care about anything else. Right, so we need to have both tools and algorithms to actually do these very specialized assembly tasks, but also figure out how to formalize them so we can actually validate what the output of those things are. And also, as we kind of go forward with all these approaches, we really need to think hard about, well, how we validate things, and also what are the standards that we want to impose on a community. So with a single genome, it's very simple. I want to give you one chromosome, right, or one FASTA file. That's the output. Do we want the metagenomic data to get a single FASTA file? Do we want to have each sequence in a FASTA file with an annotation saying, I think this is a 10-copy thing, or this is a 100-copy thing, and so on, right? Do I want to actually cluster these somehow based on saying, well, I think these are roughly the same abundance, or these are roughly the same GC, and all this kind of stuff? And we don't have good standards of figuring out what type of information we want out of the data. And I don't think we all agree what type of information we want, but I think where the community really needs to go is actually figuring out what these use cases are, and figuring out how to develop standards for the use cases and develop algorithms for specific use cases, rather than just saying, here's a new metagenomic assembler and it does better than the other ones. Because I don't, you know, I'm not sure that any one metagenomic assembler necessarily does better than all the other ones, but it might do better on specific tasks that are still important for biologists. Okay. So I think that's all I have. I want to leave some time for questions. I also want to acknowledge a lot of people who work with me and the funding agency. So thank you very much. Yes?
So I'm all in favor of using likelihood scores as sort of ways of evaluating things, but the likelihood score is only as good as the model.
Yes.
So you've really hidden something under the mat there.
Right.
You know, it's worthless if the model is wrong.
You're absolutely right, and we actually saw that. Essentially, with the wrong model, you can get assemblies that are better than the truth, and that's, yeah. But I would even there. I think I would like more transparency in terms of this is what my likelihood score measures, and there's also a lot of room for improvement in terms of you can model everything about sequencing process, but it's not clear that all the model is necessary to really separate assemblies, for example. So.
Can you explain a bit more about the model that you used for?
Right, so our model essentially assumes this uniform sampling from the genome in terms of the reads. You can modify that to take into account biases, but we didn't. And we also required that the mate pairs are roughly the right length apart. And we technically model sequencing errors, but we don't do the full needle non-Wunsch. I mean, we did to see how it does, but essentially we just take something like a bow tie alignment, and then based on that we compute the likelihood score for that read being placed at that position in the genome, rather than looking at all the other places where the read could go. And I mean, there's a lot of simplifying assumptions from that point of view to make it tractable. But so that combination of parameters works fairly well, and that's roughly the same thing that AL and CGAL do. But for all of those, and it's very open as to whether you start playing with the parameters and add more parameters to your model, whether that would help. One big part that's missing, and it's not in terms of, I don't know if Newton folks are interested at all, but essentially one part that we kind of swept under the rug is that if you do Bayes' rule to say to go from the probability of a read given the assembly to have the probability of the assembly given the set of reads, you need a probability of what an assembly, well, probability of the assembly itself, right? You need some kind of prior understanding of what the genome should look like. And we don't know how to model that. So essentially we say all the assemblies are equally likely biologically. And that's obviously not true. But we don't really know how to model the genome itself. You can also need to model the set of reads, but if you have two assemblies from the same set of reads, that number goes away, right? So, but a probability of a specific assembly is something we don't know how to model, and that would be very, very useful in deciding which assembly is better. Yeah, so sorry, I'll come back to you. So, yeah.
You were a little bit provocative with your title.
Yes.
So I'll be a bit provocative back. So if given PacBio is now quite routinely established, as you say, you can do complete bacterial genomes trivially now, and we have aluminum molecular, which looks quite good, and we know that the nanopore is pitching up in our labs in the next few weeks. Isn't this going to be a solved problem within a year or two?
For a single bacterial genome, probably yes. For a metagenomic sample, I mean, I'm not sure 10 kb reads, even if they're perfect, will allow me to separate out haplotypes of different bacteria, so to actually figure out what the strain structure is within the bacteria in a metagenomic sample. So I think that's the big challenge for metagenomics is, well, and that's a big question for I don't necessarily for this audience, but definitely what are the biological questions that you want to ask of the sample, right? So essentially, if you just want to know, if you want to answer a very simple ecological question, how many, are there lots of proteobacteria or something like that in my sample, you don't need to separate out strains. But for a lot of clinical applications, I think you do need to separate out strains. And obviously, and it's pretty clear if you look at viruses and so on that people do want to separate out the different strains. And,
I agree with that, yeah.
And I think
haplotype reconstruction will be a lot easier with 10K.
Would be a lot easier, but I think it's still far from being solved even at 10K. I think you really need to go to 100K, 200K, and stuff like that. And that's depending on the specific application that we have. That's my biased view, but yeah.
Another question?
Yeah, so I had a meeting with some medical people last week, and they're studying antibiotics and resistance genes, and they're very keen on doing assembly,
right?
And they're encoding genes. After Focus talk yesterday, I didn't think that was going to be particularly feasible. So, but then your talk, you said actually assembling the genes works reasonably well. So would that be a
Yeah, so I would say assembling the genes works reasonably well. The question is, and that's probably why a lot of problems come in, so in theory that's true. In practice it's not exactly clear whether the specific assumptions that a particular assembler makes are going to avoid or make mistakes in genes. So like a very typical example would be you have high enough variation within the set of genes that essentially the assembler would say, I'm just going to break inside this gene, even though there are only a couple of SNPs here and there, right? So you probably do want to design an assembler that specifically just wants to assemble the genes.
And
right?
So they're going to look at 200 people, 3 time points each, and I think it's going to be about 18 billion reads.
Right.
And what would your strategy be? I mean, obviously you can't assemble all of those, but would you take one person and make a reference out of that, or would you?
I don't know what the right answer is. I would actually, right now, if I were to do it today, I would assemble each person separately, and then use some of these information about correlations or maybe even links to actually combine the assemblies of multiple samples into one. When the very least, just to find the one gene in one assembly that now you find in other ones. Yeah.
Another question in the
Yeah.
back.
So if we follow your logic, what do we do with all the accumulated data, and how do we evaluate what's good, what we keep, what we throw away, or do we just start all over again?
I don't know. There's been talk at NCBI, for example, just go through all the through all the genomes in the database and try to figure out if they're correctly assembled or not. And I don't think anything ever happened with that. But I would, what I would like to see is more assemblies also come with flags along the assembly saying, I think there's a mistake here, or there could be a mistake here. And we used to do this, right? And we tried to convince the community to do this more seriously. So the NCBI at one point, you could get the assembly and flags along the assembly and where all the reads went into the assembly, so you can actually validate it yourself. But then next gen came around, and essentially all of that disappeared. But I would really want genome assemblers to actually tell you not just the FASTA file, but say, well, I think this region might be wrong. I'll give it to you anyway, but be warned, and have that annotated just like you annotate the gene. And I don't see why we couldn't do that. But with what exists, it would be a pain to go back and fix it.
Okay, we're going to take 2 more questions.
Yeah.
Wally and then...
Yeah.
So the first comment is a trivial one. I mean, if you don't know, if you really have no prior information on which way the assembly should look, then essentially it's a uniform prior. So you can
Right.
call your likelihood a posterior
Correct, yeah.
distribution if you wish to do that. And there's an advantage in that because you can then sort of just use the general sort of Bayesian thinking,
Right.
Including the possibility of putting posterior probabilities on a whole, instead of on single solutions,
Right.
on solutions which are really sets of solutions. So you actually showed a little graph there where you had some, you said, is it okay that we could have some sort of mixing up of haplotypes
Right.
as you read through? Rather than say, here's my assertion as my best guess, you can actually embody a great number of possibilities, and so you can actually then attach a posterior probability to that entire thing. So these things are not sort of necessarily. You don't really need to invent a new paradigm.
Right.
Really, the Bayesian thinking would allow you to do all of this, and with the possibilities of sort of putting graphs as solutions as opposed to a sort of,
Right.
genome sequence, then you can actually assess the posterior probability on that
Right.
on that graph and the accuracy of that. So then that leads you on to the possibility of, which I think you've kind of been hinting at, but didn't explicitly mention, that you could simply actually have a loss function, and the loss function would depend on actually what you want to do this for.
Right.
And so different people would have different loss functions depending on their question of interest. So I think you could probably set this whole thing up within that paradigm, but it doesn't change what you do, but it would actually allow you to express all these things with a sort of coherent sort of probability thinking behind it all.
Yeah.
Just a comment there.
Yeah, no, thank you, yeah. Now, I do think of with metagenomic assemblies, we do want to have a graph as the output rather than a FASTA file, for example, that would start allowing you to do a lot more interesting things with the data. So.
Did you have a follow-up,
Okay.
or that was it? Okay. One more
Okay,
question?
Alice.
Yeah, I was just wondering, in your likelihood score, even if you can't reconstruct haplotypes, if in addition to the sequencing errors, you can somehow incorporate SNPs into.
Can you repeat the question?
Can you,
Yeah.
if in your consideration of the likelihoods, if you can distinguish between SNPs and sequencing errors, so because that, you know?
So,
Repeat the question for the audience, because not...
yeah. So the question is, when you do the likelihood scores, if you can distinguish actual SNPs, like actual polymorphism in the data from sequencing errors. So in theory, yes, we have not done it for when we did metagenomic assembly. We still treat them as at some level errors. But philosophically, I think that if an assembly that completely separates out the haplotypes should give you the best likelihood score, and that's what would happen if you just treat them as sequencing errors.
Yeah, but maybe you could come up
But that's
until you have to find when you have haplotypes, so sort of treat it.
right. So you should be able to do it. I mean, assuming you can distinguish them in the first place, you should be able to score them differently. Yes.
Okay. Thank you very much.
Thank you.